Welcome back to the Cambridge Neuroscience Interdisciplinary Seminar Series. This series features current work from the neuroscientists across the schools and departments of our university, reflecting the pioneering work and diverse interests of our members here. Each seminar focuses on one of the new interdisciplinary themes of Cambridge Neuroscience, which we will be launching later this year. For more info on the talks covered in this seminar series and all things neuro-related in Cambridge, please follow us on Twitter at CamNeuro and follow the links below. So, welcome everybody to today's Cambridge Neuroscience Interdisciplinary Seminar Series, and I'm really delighted to welcome Professor Angela Roberts here today. Angela is a professor of behavioural neuroscience at the Department of Physiology, Development and Neuroscience here at Cambridge. She actually studied at Cambridge. She studied her PhD in neuroendocrinology and was a lecturer in the original anatomy department before coming to PDN. So without further ado, I'm going to pass you over to Angela and hear her talk. Welcome, Angela.
Thanks, Dervila. Okay, well, thank you everyone for coming along. And so as you can see, the title of my talk today is Dissecting the Neural Circuits Underlying Prefrontal Regulation of Reward and Threat Responsivity in a Primate. So the focus of the research in our laboratory for many years now has been a systems approach to the study of the prefrontal cortex in a non-human primate. And we're particularly interested in the role of the prefrontal cortex in regulating positive and negative emotions. And we hope ultimately that our findings will provide us insight into the etiology and treatment of emotional disorders, which are prevalent not only in, for example, psychiatric conditions such as anxiety and mood disorders, but also neurodevelopmental disorders such as autism, and also neurodegenerative disorders such as Parkinson's and Huntington's disease. But in today's talk, I'm going to focus very specifically on one part of the prefrontal cortex, the ventromedial prefrontal cortex, and consider its role in two major symptoms of depression, namely anhedonia, which is the loss of pleasure, and also anxiety, which is highly comorbid with depression. So to start with then, if we just, I can't see how I can move on now, hang on. Yeah, if we just start considering the depression itself. So I've just got something in my way that I'm trying to move because I don't quite know how to do it. How do I move this out of the way? Oh, yes, here we go. Brilliant. I'll put that over there somewhere. Brilliant. Okay. So when we think about depression, one of the first things we should recognize is that two patients can be diagnosed with depression but have no overlapping symptoms. Even if they have the same symptom, it's very likely that the psychological neurobiological causes are not the same, since the clinical symptoms themselves, such as anhedonia and anxiety that I'm going to talk about today, are often poorly characterized. Oh, goodness, sorry, I'm still having trouble trying to turn this thing on. Yes, here we go. The next thing to consider is even when treatments do work, we don't know how. So we can't predict which treatment will work best in any given patient. And so for all those reasons, we need better stratification of depression in order to develop these more personalized treatment strategies. At the core of depression is dysregulation of prefrontal function. Now, what I'm showing you here is a sort of schematic of many of the brain regions that we know are involved in emotion regulation. And we've learned a lot about the subcortical circuits that are important for positive and negative emotions from studies in rodents. So, for example, we know about the importance of the amygdala, the bed nucleus of the stria terminalis, and its projections down into the hypothalamus, to the brainstem, for controlling specific emotional states, both positive and negative. But we know far less about the role of the prefrontal cortex itself, shown here in blue, which we know is dysregulated in depression. And this is just to highlight the many different heterogeneous regions we know exist in the prefrontal cortex. So here's a lateral view of the prefrontal cortex, a medial view and the orbital view. And you can see there are many distinct cytoarchitectonic regions within the prefrontal cortex, which we think ultimately do have distinct functions. And so when patients with depression have been studied, especially using functional MRI and also structural MRI, changes in activity have been found not only up in the lateral regions of the prefrontal cortex, but also in various aspects of the medial prefrontal cortex, including this ventromedial prefrontal region here that I'm going to focus on today, and also the orbitofrontal cortex, both lateral and medial regions. But we have very little understanding of how these different regions of the prefrontal cortex ultimately contribute to the myriad of symptoms associated with depression, whether it's anhedonia, sadness, despair, or, as I've said, this sort of comorbid symptom of anxiety. And so we've been focusing particularly on two of these symptoms, anhedonia and anxiety, to try and get a handle on exactly what is the role of the distinct regions of the prefrontal cortex and how that might help us understand depression. So like I said at the start, we've chosen to work with a New World monkey, the common marmoset. And the reason we've done this is because when you want to study the cortex, in the human, over 80% of the brain is given over to the cortex. And the cortex is, of course, the powerhouse of the human brain. Whereas in the most popular species used for studying kind of systems, neuroscience, the rodent, only 28% of the brain is given over to the cortex. Whereas this suddenly becomes much greater once you move into non-human primates. The marmoset, 60% of the brain, and the rhesus, 72% of the brain is given over to the cortex. And when you look specifically at the prefrontal cortex and the organization of that, this shows you ventral and medial views of the human brain, the marmoset brain. And the first thing to notice is that whilst you may not be able to see this too clearly, nevertheless, the overall patterning and organization of these regions in the human brain and the marmoset are very similar. Whereas, as you can see, it's very different from what you can see in the rodent brain. And so that's one reason why we're wanting to use the marmoset to try and understand some of these complex prefrontal regions. And finally, when we first set out to study emotion, we realized that there was another gap we wanted to bridge, and that was with respect to the kinds of measures you use for studying emotion. In humans, you either ask them questions about how they feel, subjective reports, or you measure their autonomic activities, such as skin conductance response or heart rate. Whereas many of the studies in rodents, especially for studying the negative sort of domain of emotions, the measure was primarily behavioural, and it was a freezing response. And so we wanted to be able to do studies that would not only allow us to back translate to rodents and be able to make use of the wealth of evidence that had been already developed in rodents about these emotional circuits, especially subcortically, but also be able to forward translate into the clinic and be able to map our behavioural and autonomic changes to those seen in humans. And so in the marmoset, in many of the studies I'm going to show you, we, in a one-off surgery, we implant the marmoset with a telemetry probe into the descending aorta, which subsequently allows us to measure cardiovascular activity in a freely moving animal. So I've just lost my cursor again. Where's it gone? Here it is. So the three domains I'm going to talk about today are firstly negative valence, which will give us some insight into anxiety, positive valence to give us some insight into anhedonia. And then if I have time, I'll also mention some sort of basal cardiovascular activity measurements we've been taking, which actually gives us some insight into why there is such a close interaction or close correspondence between people suffering from heart disease, which are more likely than the general population to develop anxiety and depression. And likewise, people suffering from anxiety and depression are more likely than the rest of the population to develop heart disease. So there's clearly a very strong relationship between psychiatric disorders and cardiovascular disease. And so that's something else we wanted to look at. So why study the ventromedial prefrontal cortex? Well, as you can see here, this is a human brain. Here's the ventromedial prefrontal cortex shown here. And the reason we focused on it is because, first of all, activity within this region has been associated with sadness induction in normal controls. It's also been shown to be differentially activated or inactivated in depressed patients. And similarly, this same region has also been associated with successful treatment response in terms of treatment success with SSRIs or SNRIs. It's also reductions in activity in this region have been associated with successful treatment with ketamine. And of course, from some of the early work of Helen Mayberg, a psychiatrist working in this region, she developed and targeted deep brain stimulation to this region for also treating treatment-resistant patients. But the thing to remember is that this is a very complex region. So the ventral medial prefrontal cortex, first of all, gets called, it's this region here in the human, and it's composed of a range of different regions: the frontal pole, area 10, bits of 11 and 14, and then areas 32 and 25. And then separately, sort of in the more caudal region of this area, is what's often called subcallosal anterior cingulate. And so these names get used a lot in studies in humans, functional imaging studies, but actually, again, they're not referring to one particular area. There are a cluster of areas. And so we haven't really got a very good understanding of how these different regions within the ventral medial prefrontal cortex actually contribute to depression. Of course, if we can turn our attention to animals and make selective manipulations in this region within a monkey, we can actually start to tease apart what these different regions, how they're contributing to the different symptoms. And just to show you that this subcallosal region has actually been highly sort of related to successful treatment. So functional connectivity of this region with other regions in the prefrontal cortex have been associated either with successful pharmacological treatment but failed CBT, or with successful CBT and failed pharmacological treatment, or with successful treatment with transcranial magnetic stimulation in the dorsolateral prefrontal cortex, which is another treatment that's been recently introduced for treating treatment-resistant patients. But in all of these studies, significant positive or negative correlations have been found with subcallosal anterior cingulate cortex and its connectivity with other regions of the brain, suggesting this may well be a core area for understanding depression. So first of all, I just want to sort of acknowledge all the people in the lab more recently who've been involved in these particular studies. Today, I'm going to be focusing primarily on the work of Leith Alexander and Dr. Christian Wood, with Gemma Cockcroft and Lauren McIver really supporting those studies, and also, of course, my long-term collaborator, Dr. Hannah Clark, and also Steve Sawiak, who's been very instrumental in all our imaging studies I shall show you. So the first study I'm going to tell you about is work done by a very talented PhD student at the time working with us, who was a medical student, Leith Alexander, and he first started looking at the effects, of overactivation of area 25, this subcallosal region in the marmoset brain. To overactivate it, what we do, we originally were infusing a glutamate transporter blocker, dihydrochinic acid, directly into this region, which actually acts to allow glutamate to stay around for longer, as it's not being taken up by the synapse so quickly, and therefore enhances the overall excitability of this region. And so he went on to look at the effects of overactivating area 25 first on aspects of anhedonia, the loss of pleasure. So this is a word cloud of lots of different words that people use to describe their anhedonia, including no excitement, no pleasure, apathy. And all these words don't all mean the same thing. They actually are capturing different aspects of the anhedonic state, which hasn't always been appreciated or measured in studies of depression in animals. So, for example, when we're talking about the loss of pleasure, we're often thinking about it in terms of consuming rewards. So, for example, if I like to go home at night and drink a small glass of Merlot, then I may get pleasure out of drinking that Merlot. Whereas potentially if I have what might be called consummatory anhedonia, then I no longer get pleasure out of drinking that glass of Merlot. Alternatively, I may still get enjoyment out of drinking that glass of Merlot, but in the middle of the afternoon, when I start thinking about it, I don't get any pleasure anymore in the anticipation of it. And then that's what we might call anticipatory anhedonia. And of course, in both these examples, we can test it in the laboratory. We can test an animal's sort of rewarding aspects of the consummatory, their consummatory rewards. That's the sucrose preference test that's been used in humans for studying anhedonia. but it's very selective for just looking at how an animal appreciates, in this case, a sweet solution. Alternatively, using really classic Pavlovian conditioning, you can study how much an animal anticipates the excitement associated with getting access, in this case, in this study in the marmosets, with a box full of marshmallows, which they learn is predicted by a tone that comes on 20 seconds before. So that's anticipatory arousal. And if you lose that sort of anticipatory anhedonia, then that's a task you can use to measure it. And then finally, of course, there's the maybe that I kind of look forward to the glass of Merlot and I'm enjoying the glass of Merlot. But if I have to actually, before I go home, actually go around the shop to get the glass, to get a new bottle, and it's probably going to be raining and it's probably going to be a queue because of COVID, then suddenly the motivation is not there anymore. And so the other aspect of anhedonia is motivational anhedonia. And you can measure that in animals by looking at their ability to work for reward and actually make more and more responses in order to get access to their reward. And so you see how hard they're prepared to work for it. And that's how you can measure sort of motivational aspects. So with these different tasks, we then went on to look at the effects of overactivating area 25. So this is just the Pavlovian paradigm, and just to show you what we're measuring here. So this is systolic blood pressure across time. This is baseline period, and then the 20 seconds of a CS comes on, in this case a tone. And if the animals learned that the tone predicts reward, then that's what we might call the anticipatory period. And you can see there's a rise in their blood pressure. And then once they get access to the reward and they get 2 minutes of being able to eat as much as they possibly can of this marshmallow, then this is what we'd call the consummatory component. And you can still see blood pressure goes up even more, whereas blood pressure doesn't change at all when you're presented with the CS minus that predicts access to an empty box. You can also measure behavior. So this is the kind of behavior they show to a tone that predicts reward. It's a sort of series of rather agitated head jerks that the animal makes to the sound and sort of kind of excited response. And you can see they make it to the CS plus, but not to the CS minus. So that's our anticipatory behavioral response. And then we can also measure consummatory behavioral responses. We can measure how much reward they actually consume when they get access to the marshmallow, and also how quickly they start eating the food. And so this was originally set up by Ivona Wiki, a PhD student in the lab many years ago. And we subsequently later went on to use this to look at the effects of overactivating area 25. And what he found was the following. Over here, we see the blood pressure response to the CS plus and the CS minus if you infuse saline into area 25. And this is the head jerk arousal response if you infuse saline into area 25. But if you overactivate area 25 with this infusion of dihydrochloric acid, you find that both the blood pressure response and the behavioral response are blunted. So you could say it looks like we've got a sort of form of what you might call anticipatory anhedonia, a lack of excitement, a blunting of this anticipatory excitement. But there doesn't appear to be much an effect on the consummatory parts of this. So there's no significant change in the blood pressure response when they're consuming the reward, and there's no difference in the amount they consume. So it doesn't appear that the consummatory aspects are particularly impaired when you overactivate area 25. It seems to be quite selective for the anticipatory. And then later went on with a postdoc in the lab at the time, Phil Gaskin, and set up this motivational task where the animals just had to work harder and harder to get access to reward, banana milkshake. And so this just shows you the kind of exponential increase in responses they're having to make in order to get reward. And again, what we found was that when they overactivated area 25, there was a complete blunting in comparison to their baseline behavior of the motivation to get reward. So it appeared then that overactivating 25, an area in depression which has been shown to be overactivated, does actually produce certain aspects of what you might imagine could be the anhedonia symptoms, which is blunting of their motivational and appetitive arousal. But not all regions in the ventromedial PFC do impact on this behavior. So we've shown you how overactivation of area 25 produces this. We've also looked at area 32, and Lath found no evidence of either the inactivation or overactivation of 32 impacting on appetitive behavior. And But we did find another study that was performed in the lab by Eula Masudi, who looked at the effects of inactivating or overactivating area 14 adjacent to area 25, did actually produce a similar effect. But I'll show you later on that we know definitely it was not because there was any sort of crossover into area 25. It's an independent contribution to this. So we then went on to look now at the negative emotional state and to see if there was any evidence for this region also being involved in that. And this again is the work of Leith Alexander, but also he was joined also by Dr. Christian Wood. So one way of thinking about negative emotion, which I really like, is this theory sort of proposed by Fanzolo and then sort of adapted and developed more by Dean Mobbs more recently, which is the idea that threat in the environment can either be on top of you and very proximal, and you have to react quickly. So, you know, with panic at the very extreme end of one end of this, then all the way back to the other end where you may be worrying about something that never happens. And that's where you may be anxious about something, but in fact, it may never happen at all. And what Dean Mobbs has suggested is that you could imagine that there's different kinds of sort of executive processes engaged across these different sort of threat in time and space, so that when you're up this end in the sort of immediate threat condition, you haven't got time to think, you just have to react. Whereas right down this end, you've got all the time in the world to think, so you can engage as much of the prefrontal cortex executive processing as you like to try and sort out what this thing is you're worrying about. So he suggested that you may well see a lot more prefrontal contributions to this end of threat, the anxiety end, rather than this fear end. Well, anyway, you can measure these different aspects of threat in time and space in a range of different tests. Pavlovian conditioning is what you might call this stage, sort of post-encounter threat, sort of cognitive fear, as they've called it. We also have used another test, the human intruder, to measure something which is much more in a situation where the threat is more uncertain, and is what you might call encounter anxiety. And I'll go on to describe that in a minute. So using these two tests, Leith went on to look at the effects of overactivating area 25. So the human intruder test is a test we've used a lot now in the monkeys. And in fact, it was originally set up and developed by Yoshiro Shiba, a PhD student in the lab at the time, along with Dr. Andrea Santangelo, and also more recently, Sean Kwa. And they studied all the different behaviors the marmosets show, and they show a range of behaviors to the human intruder. You know, when they see the human intruder, they don't know whether they're going to get food. It's an unknown person to them, so they don't know whether they'll perhaps get fed, or perhaps they'll try and catch them. So it's just uncertain kind of time for them. And so what happens is that marmosets, you know, depending on how reactive they are, they jump to the top of the cage if they're scared. They'll go more to the back of the cage as well if they're more anxious. They make these vocalizations. They also, of course, you can measure how still they are, and they also show these species-specific sort of head and body bobs. And basically what Yoshi and Andrea at the time found was that you can put all of these behaviors into a kind of principal component analysis, or more recently, Sean's used a exploratory factor analysis, and show that sort of these different behaviors are related to one another, and they all sort of weight upon one factor, which looks like some kind of emotional reactivity factor. Because if you score high on this score, you're at the top of the cage, at the back, doing lots of head and body bobs and making quite a lot of vocalizations, and you're relatively still. So you look like you're relatively quite anxious. And so Leith looked at the effects of overactivating area 25 in these animals on this task. Here's the EFA score. The higher up this axis you are, the more anxious you are, or the more emotional reactive you are. And what he found was as soon as you overactivate area 25, they show a lot more of an emotional reactive effect. And they're at the back of the cage doing a lot of head and body bobbing, looking worried. He then went on to look at Pavlovian conditioning. And so the animals have been trained up to learn that a CS- predicted nothing, and a CS+, another tone, predicted an aversive loud noise. And this shows you the control conditions, and this is their cardiovascular activity. And you can see nothing happens when the CS- comes on because it means nothing. When the CS+ comes on, normally you do start seeing a rise in the cardiovascular activity, which then rises even more when you're presented with the aversive loud noise. And also there's some darkness here as well. They're plunged into darkness for about 20 seconds or for a couple of minutes. And there's this intermittent noise, which they don't like. And anyway, you can see a large rise there. And then very rapidly, it comes back down as soon as the noise goes off and the darkness goes, and then it stays flat. If you overactivate area 25 one session, you find that straight away, the animals are showing an enhanced cardiovascular blood pressure response all the way through the session is the main thing. They still show a bit of an additional rise to the CS plus and up through to when the US comes on, but then they're also much slower to come back down from the US. So once the darkness is gone and the aversive loud noise is gone, these same animals, when they're being given saline, their responding goes down quite rapidly and they sort of recover from that, but they don't recover so much when they've had overactivation of area 25. So in summary then, it appears that both the sort of Pavlovian conditioning expression, there seems to be this overall generalization of the conditioned response when you overactivate area 25, and they're generally more anxious both to the context, regardless of whether the CS minus comes on or not, they're showing this heightened reactivity. And just to show you, you don't get this with every manipulation you ever do in prefrontal cortex, this just then shows you, with all the studies we've now been doing, we found that only area 25 and this anterior cingulate reading in area 24 actually has an impact on both heightening reactivity to conditioned threat, as well as heightening reactivity to uncertain threat. The other prefrontal areas we've looked at, including 11 and 13 and 14, have no impact on conditioned threat, but they do have an impact on uncertain threat. So when you're in a more sort of distal threatening condition, it's more uncertain, then it does seem that many regions of the prefrontal cortex get engaged, and dysfunction in any of these regions will disrupt the animal's reactivity to this human intruder, and it actually can heighten their reactivity. But when you come to the Pavlovian conditioned proximal threat, most of these prefrontal areas aren't involved. The only areas involved are 25 and the anterior cingulate, the sort of older, more less differentiated regions of cingulate cortex, not the higher order executive areas, which would match what Dean suggested might be the case from his imaging studies. So focusing in on area 25, the last thing Laith did with another student in the lab, Chloe Wallace, who was a PhD student with Hannah Clark at the time, was they looked at the effects of both overactivating and inactivating area 25 on basal cardiovascular activity. So this is when animals are in the test apparatus, there's nothing going on, there's no reward, there's no punishment, they're actually quite bored, but it's kind of a neutral condition, and you measure their blood pressure and heart rate variability. And what we found was if you overactivate area 25, that actually you increase heart rate, you reduce heart rate variability, which means you make the heart rate less flexible, which is very much characteristic of patients suffering from anxiety and depression. They also show reduced heart rate variability, and you tend to push the balance of the sympathovagal system towards the sympathetic fight and flight system. Whereas if you inactivate area 25, which is what Chloe looked at with Hannah, you actually find the opposite: reductions in heart rate, increases in heart rate variability, so you're actually more flexible, and if anything, you push the balance of the sympathovagal towards the vagal, so towards more the resting state. So, and again, just to highlight, this function of area 25 in regulating basal cardiovascular activity is not seen by any other prefrontal area. We've looked at other areas in ventromedial prefrontal cortex. We've looked up in area 24. We've looked across in the orbitofrontal cortex. None of these areas have any impact on basal heart rate variability. It's only area 25. So in summary then, from those original studies, what we found was that overactivating area 25 not only enhances the overall negative emotional state of an animal, it pushes the basal cardiovascular activity towards the sympathetic system. It causes generalization of conditioned threat, and it also enhances reactivity to more distal uncertain threat. But of course, remember, it's also completely blunts anticipatory and motivational reward. The two key symptoms often found in depression in an area which we know is overactive very often in depression. So our next question was, well, if Area 25 is doing this and producing these two quite distinct symptoms, is it doing it through the same or different downstream pathways? So we know the Area 25 projects out to the BNST, the bed nucleus striatum terminalis, to the amygdala, the ventral striatum, and the hypothalamus. So we wanted to find out, what areas are being engaged when we overactivate Area 25. And so the first thing we did was use FDG PET, fluorodeoxyglucose PET, to allow us to look at the circuits that may be involved when we're overactivating Area 25. So what we do is we implant the animals in a one-off surgery again, where we actually implant a tube into the jugular, but the tube is connected to a plastic import that's placed underneath the skin. So on the day of the actual injection, all we have to do is inject the animal under the skin, a subcutaneous prick of the radioactive ligand, in this case, fluorodeoxyglucose, but then that goes directly into the jugular. And then the animal is basically placed in an environment for 30 minutes where they either are in an environment which is threatening, or they're placed into an environment which is appetitive and they're anticipating reward. And after that 30 minutes, we take them out the apparatus, they get anaesthetized, put in the scanner, and we measure FDG uptake. And basically, the amount of FDG uptake you measure is a proxy measure of activity. And those areas of the brain that were particularly active in that half an hour preceding, when you first put the FDG on board, if they're more active, they take more FDG up. So you can see which areas were most active. And what Leith and Christian found was that if you overactivate Area 25 in an appetitive context, so the animals get put into an appetitive environment, then not only do you see FDG uptake in Area 25 itself, which we hope we do, because that's where we did the actual overactivation, but we also see increased activation in the dorsal medial prefrontal cortex, in the insula, and also we see some reductions in activity down in the brainstem, in and around actually some of the serotonergic brainstem neurons. So that's what happens in an appetitive context. It's a rather different network that's activated or engaged when you overactivate Area 25, but this time put the animal in a negative context. So now the animals have been overactivated with 25, but this time they've been put into a negative context with stimuli that predict aversive, sort of darkness and aversive loud noise. Then what we find is that, again, not only is there overactivation of Area 25 itself as measured by FDG uptake, but there's increases in the amygdala, in the insula, and the hypothalamus, which is exactly what you'd expect if you were making an animal more anxious. But really interestingly as well, there is a reduction in activity in this region here, which is the dorsolateral prefrontal cortex, area 46. There's also a reduction in this ventromedial, this ventrolateral region of the caudate nucleus. And this is interesting because in depressed patients, not only is there overactivity in Area 25, but there are reports of reductions in dorsolateral prefrontal cortex, which is why one of the treatments for treatment-resistant depression is in fact transcranial magnetic stimulation in dorsolateral prefrontal cortex. So it looks like all you have to do is if you just overactivate Area 25 selectively, you actually cause reduction in Area 46. So having shown there's a different network involved when an animal's in an appetitive or an aversive context, we wanted to then ask the question, so are there distinct pathways by which Area 25 is producing these distinct symptoms? And this is the work that Dr. Christian Wood has been really forging ahead over the last couple of years. So these viral mediated vectors were first brought into the lab by Johan Alcio and Andrea Santangelo to sort of see if we could start getting them to work in the marmoset. And then Lydia and Leith, in collaboration with them, started to try and look to see if we could use dreads to overactivate Area 25. Christian has taken that one step further and started to take the dreads and actually start to look at pathway-specific manipulations. And so basically what Christian's done is he's taken AAV8 virus, linked up to CAM kinase 2, which targets pyramidal output neurons in the cortex, and the DREAD construct, which is a sort of reworked muscarinic receptor, M3DQ, which and of course, DREAD stands for designer receptors exclusively activated by designer drugs. So this is the designer receptor, and it's exclusively activated by a designer drug, the designer drug being clozapine N-oxide, CNO. And when CNO activates this designer receptor, it enhances neuronal excitation. And so what Christian's gone on to do is he's targeted area 25 with this DREAD-containing AA8 virus. And then in the first instant, we're wanting to go on to use CRE for being able to do CRE-dependent pathway manipulations. But in this first instant, what Christian's done is he's cannulated the nucleus accumbens and he's cannulated the amygdala. And so what he's able to do is, having placed the DREADs in area 25, normally you'd give the CNO systemically because it would activate the designer receptors everywhere in the brain that have come from area 25 where you've placed them. But in this situation, he's not doing that. He's actually placing CNO through these cannulae, either to target the nucleus accumbens, which means he's targeting the 25 accumbens pathway exclusively, or he's put the CNO into the amygdala where he's targeting the 25 amygdala pathway exclusively. And he puts the CNO into these cannulae, and then after 30 minutes, he looks at the effects on these appetitive, reward, and punishing paradigms. And this just gives you a little bit of a feel for the kinds of expression that Christian has been able to get within area 25 here, with the targeting these output projection neurons. So what did he find? So he wanted to ask the question, which pathway, for example, may be responsible for the blunting of anticipatory arousal produced by overactivating area 25. We know area 25 projects the accumbens. We know the accumbens is very important for anticipatory arousal. So what happens if you actually overactivate that 25 accumbens pathway? And so what he found was actually, if you place CNO directly into the accumbens, you actually blunt the behavioural arousal associated with the CS plus compared to saline or vehicle, and you also blunt the blood pressure arousal associated with the CS plus. And so the CNO directly into accumbens blunts this in the same way as when we overactivated 25 in general. But interestingly, when he placed the CNO directly into the amygdala, so now he's targeting the 25 amygdala pathway, there's no such blunting. So no effect on the head jerks, which, there's lovely head jerk arousal, there's lovely blood pressure arousal, even though CNO has been put into the brain, but that's because it's been put into a pathway which presumably is not producing that effect. He then went on to ask the question, so if the area 25 accumbens pathway is responsible for the flattening of the appetitive arousal, what's responsible for the heightening of the negative arousal in the human intruder test? And so here he's looked at the performance of the human intruder test, this time again giving CNO either into the nucleus accumbens or into the amygdala. And this time what Christian's got is the opposite effect. Now he finds that if he puts CNO directly into the amygdala, if you're looking at increased threat reactivity, it's going this way up the axis. So this is a relatively chilled animal, low down on the axis. When you put CNO into the amygdala, you heighten his reactivity to the human intruder, but only in the amygdala. If you place CNO into the accumbens, it has no impact. So what Christian's been able to show is that whilst when you overactivate area 25 in general, it enhances both the reactivity to threat and blunts the reactivity to reward, actually, these two effects are happening through two different separate independent pathways coming out from area 25 to the accumbens versus area 25 to the amygdala. So finally, I just want to give you a glimpse of some of the treatment effects we've been starting to try and look at. So you remember I showed you earlier on that the subclosed anterior cingulate cortex appears to be related in patients in terms of the functional connectivity of it to a whole load of different treatment outcomes. So we wanted to test that in the marmosets. And so We know patients, some can be treated with SSRIs and it can be quite successful, but we know that many are non-responsive. We also know that those that are responsive to SSRIs often show residual reward processing deficits, i.e. the anhedonia deficits. But more recently, an alternative drug, ketamine, which is a rapidly acting drug, has been found to actually have quite long-lasting effects and apparently on the anhedonic aspects of the depressive syndrome. So we wanted to look at the effect of ketamine in our marmoset model. So originally, in the original model, going back to when we used to do dihydrochloric acid, the pharmacological way of overactivating 25, what Laith did was he took animals at time 0, he gave them ketamine. I should point out that ketamine in patients takes somewhere between 1 and 4 hours to have its action, and it's definitely fully treatment successful within 24 hours. So he gave ketamine at time 0. He then overactivated area 25 at 4 hours, at 24 hours, 7 days and 3 weeks. And each time he overactivated area 25, he looked at their anticipatory responses in the test apparatus. He did it with a second set of animals, but this time he looked at the anxiety effect. So this time he gave ketamine at time 0 and then tested them in the human intruder test to look at their heightened reactivity to anxiety, to the human intruder, either at 24 hours or 7 days. And what he found was that DHK induced anhedonia was ameliorated by the ketamine given 24 hours before. So here's ketamine here. At 4 hours, DHK completely blunts the anticipatory arousal. There is no arousal. But at 24 hours, DHK now doesn't have any impact on the arousal. They still show a normal anticipatory arousal, even though they've received overactivation of area 25. But this is having had ketamine 24 hours before. And this effect is still seen 1 week later, after just 1 injection of ketamine, but it's gone by 3 weeks. But interestingly, it's not the case for the anxiety. Overactivation of area 25 with the DHK produces the heightened anxiety score. If you give ketamine and then look at the effects of DHK 24 hours later, there's no effect. It's the DHK still produces the enhanced anxiety. It's not ameliorated by ketamine, and it's not ameliorated 7 days later either. So finally, in the last experiment, Christian decided that he would really like to start looking at where ketamine may be having this effect on ameliorating the anticipatory anhedonia. And so he overactivated area 25, this time with the dreads, but this time he gave the CNO systemically. So he was just generally causing this area to be overactive, not a selective pathway, just a general area. But then he gave ketamine 24 hours earlier, either down, well, no, actually down the nucleus accumbens probe. So nucleus accumbens received ketamine 24 hours before, and he looked to see whether that would have an ameliorative effect on the overactivation of area 25 24 hours later. And the answer turned out to be yes. So if you give ketamine 24 hours before, directly into the accumbens, you block the flattening of anticipatory arousal, appetitive anticipatory arousal, normally produced by systemic CNO, which of course you can see is caused by systemic CNO if you only inject saline down the accumbens probe. So, and the effect was seen whether you look at blood pressure or behavior. So in summary then, what I would like to try and show you really is that, well, I think we've seen that by overactivating just this one area, area 25, we can produce, first of all, multiple symptoms associated with this one single etiology, effectively heightening the threatening responsivity and blunting appetitive arousal. But we've also been able to show that, in fact, these two symptoms are dependent on distinct networks. And blunting of anticipatory arousal is dependent on area 25's projection to the ventral striatum, the nucleus accumbens, whereas its heightening of arousal is dependent on its projection to the amygdala. And then more recently, of course, now Christian's shown how ketamine can actually act directly into the ventral striatum to ameliorate the anticipatory arousal effects. And finally, it just leaves me to thank all the colleagues over the years that have contributed to this work in the Innis building, our primates, and these are our marmosets. Thank you.
Thank you, Angela, for that wonderful talk and a real guided tour through your research. Join us next time when we welcome Dr. Yara Erez. Formerly of the MRC Cognition and Brain Sciences Unit, Yara has recently started her own lab in Bar Ilan University in Israel. Her multidisciplinary research focuses on neuroimaging of the human brain to identify and understand functional brain networks. Her group's research is in the intersection of neuroengineering and cognitive neuroscience, and they use multiple imaging modalities to understand brain function. She will be speaking to us on cross-modality imaging of the neural systems that support executive functions. For more info on this seminar series and all things neuro-related in Cambridge, follow us on Twitter at CamNeuro and follow the links below. See you next time.